Hello and welcome back to the second part of our two-part series about Beam Therapeutics. If you haven't seen my first part, I highly encourage you to watch it. I leave a link here for you, as well as in the description below. In the first part, you'll learn why gene editing is becoming increasingly popular in medical practice and has the ability to provide handsome rewards to investors. You'll also learn about the key actors and players, inventors in the field and in this industry. And all that background information will prepare you to fully grasp and understand why Beam Therapeutics is in fact so unique in the industry. Also, make sure you stick around until the end of this video, where I reveal a special bonus for the subscribers and viewers of my channel. So let's get right into it. Although I have a PhD in biomedical engineering and over 15 years of working experience in the healthcare industry, I do not intend to give you investment advice. Please do your own research before making any investments. You are watching Health Wealth. If you're new to this channel, welcome. If you're not, nice to have you back. The bacterial defense mechanism against invading viruses, the CRISPR-Cas system, works with a guide RNA, here shown in green, in order to identify and target, in fact bind, to the desired target site on the DNA. Once the guide RNA binds to the target site, the CRISPR-Cas system is able to induce a double-stranded DNA cut. For good reason, cells have developed their own mechanisms in order to repair any damage to DNA, including double-stranded breaks. These natural repair pathways can lead, for example, to non-homologous end joining, which means that there could be additional deletion of base pairs that could lead in the end to gene inactivation. Also, an entire DNA sequence could go missing in the repair process. And finally, homology-directed repair. This means that a DNA template could be inserted through the CRISPR-Cas system if the guide RNAs are designed in such a way that they match exactly either side of the cut sites. The problem is that double-stranded DNA breaks are quite difficult to repair, and the repair process is prone to errors. You may think of some of these errors as simple genetic natural variations, but also you may think of it as cancer that can develop as a result of incorrect DNA repair. And I guess you do not have to have a PhD in genetics in order to intuitively understand that in comparison to a double-stranded DNA break, a single-stranded break should be, and in fact is, much easier to repair for the cell. And up until now, the CRISPR-Cas system has always only been able to introduce a double-stranded DNA break. In part 1 of this video series, we had the who is who of the gene editing industry. So let's expand upon that, and let me introduce you to Professor David Liu. It seems that Dr. Liu's lab is quite innovative when it comes to gene editing. What is called base editing was invented in his lab in 2016, and within only 3 years, in 2019, prime editing was invented there as well. Dr. Liu, as well as Dr. Zhang, who we already met in the first part of this two-part video series, are co-founders of Beam Therapeutics. But Dr. Liu was also co-founder of Editas Medicine and Prime Medicine. So let's take a high-level look at the science behind base editing in order to understand why, as an investment opportunity, Beam Therapeutics may provide a quite unique solution. In short, base editing involves a modified CRISPR and guide RNA component, which is modified in such a way that it is not cutting or introducing a double-stranded DNA break. In fact, base editing induces only a single-stranded break in the DNA and a combination with a base editor such as deaminase allows to change single base pairs in the DNA. For example, base editing can induce a change from C to T or a change from A to G. Just like in the standard or traditional CRISPR-Cas system, the guide RNA provides the specificity or the targeting of a particular site where the editing is intended to occur. As alluded to earlier, single-strand DNA breaks are much easier for the cells to repair, and therefore are also less prone to errors. So, base editing comes with certain advantages, such as the precise, predictable, and efficient way to edit genes. Then there's no need for a template-based homology-directed DNA repair. And the lack of double-strand DNA breaks avoids the unwanted insertion, deletions, or other large-scale genomic rearrangements that are generally associated with the repair mechanisms for double-stranded DNA breaks. So even though base editing seems to provide a much more precise way of editing the genome, as potential investors, we also need to basically find out what the addressable disease market is that this technology can potentially target. It appears that amongst the 50,000 known human genetic diseases, most are actually caused by so-called point mutations. That is, there is a swap of a single base pair in the DNA that is incorrect, leading to manifestation of disease. According to Dr. Liu, the adenosine and cytosine base editors can address roughly 60% of those point mutations which lead to pathogenic disease. So, apart from the time, of course, to validate and go through the different phases of clinical trials, the market opportunity clearly is huge. So, how do Beam Therapeutics actually stack up in the clinical trial space against big players and established players such as Crispr Therapeutics? Let's take a look. Here is an overview taken directly from Beam Therapeutics' website about their clinical pipeline. First off, we can see that Beam Therapeutics employ a number of different delivery methods for their engineered CRISPR-Cas system. And all of the clinical activities are at best in the IND enabling phase, which actually even precedes phase 1 clinical trials. And this, of course, is not unexpected due to the relatively young age of Beam Therapeutics, who were only founded in 2016. Their most advanced programs are called Beam 101 and Beam 102. And we also notice two familiar disease types, which we have also encountered by looking at Crispr Therapeutics pipeline. And these diseases are sickle cell disease as well as beta thalassemia. The fact that we see multiple companies targeting the same diseases is probably testament to a large market, as well as this being probably one of the lowest hanging fruit. So here we have now the two companies, Crispr Therapeutics as well as Beam Therapeutics, in a race trying to find a treatment for sickle cell disease. Sickle cell disease is a devastating disease. The hemoglobin in the red blood cells are malformed, and therefore the red blood cells have a sickle shape. Patients suffer a lot of pain and need frequent blood transfusions. The quality of life of patients is severely impacted, and due to frequent hospitalizations, blood transfusions, the cost of care is tremendous. The aim of the treatment pursued by Crispr Therapeutics and one of the two Beam Therapeutics studies is in fact to activate fetal hemoglobin production. It is well known that the onset of sickle cell disease is not immediately at birth, but within the first 6 months following birth, when in fact the transition is being completed from production of fetal hemoglobin to adult hemoglobin. Patients who carry the genetic malformation for sickle cell disease in fact still produce correct fetal hemoglobin and therefore until this transition takes place have no disease symptoms. So the bottom line is the treatment pursued by Crispr Therapeutics and one of the Beam Therapeutics studies for the activation of fetal hemoglobin is providing essentially a very good patch over the disease, but in fact not a true cure. There's a very good reason why we start out our lives as fetuses with fetal hemoglobin. And while, if successful, if fetal hemoglobin can be reactivated in the adult, can certainly alleviate the most severe of disease manifestations of sickle cell disease, there's also a downside for the adult who has fetal hemoglobin activated again. The developing fetus in the uterus requires oxygen just like we do. But since the fetus does not have direct contact to the ambient air and therefore cannot directly breathe, it needs to obtain the oxygen for all the metabolic activities from the mother's blood. So the transport of oxygen through the placenta strictly becomes a question of diffusive mass transport. And if both mother and fetus had the same type of hemoglobin, let's say adult hemoglobin, this would mean that inevitably the fetus would always have a slightly lower blood oxygen level than the mother. But fetal hemoglobin has a higher affinity for oxygen and therefore is better at carrying oxygen from the mother's blood through the placenta into the fetus. And with this neat biological trick, the fetus has roughly the same blood oxygenation levels as the mother's blood, although there's diffusive mass transport in between. The downside of fetal hemoglobin is that due to the higher oxygen affinity, it also is less likely to give up the oxygen into the cells and tissues that need it. This, however, is not really an issue because the developing fetus as well as young babies generally have what you could describe as a rather sedentary lifestyle. And this is in stark contrast to the adult lifestyle where we move around, we may want to do sports, be active, etc. And here fetal hemoglobin clearly carries a downside. Don't get me wrong, I am sure that any sickle cell patient would rather gladly take fetal hemoglobin as an option for treatment rather than suffering through the ongoing, ever-returning blood transfusions. But still, as I said, this is not truly treating the disease. It's merely a very good band-aid over the disease. The transition from fetal hemoglobin to adult hemoglobin happens in the young developing body due to the silencing of the expression for the gene that codes for the production of fetal hemoglobin. And this is precisely the area of the genome that is being targeted by Crispr Therapeutics and one of the Beam Therapeutics studies. The activation of fetal hemoglobin, in fact, is trying to be achieved through the CRISPR-Cas system, where the non-homologous repair mechanism is being activated at a very specific site with the hopes that the gene inactivation of fetal hemoglobin is being destroyed. And this results in fetal hemoglobin gene expression being reactivated. So with this type of treatment approach, the genetic mutation that is causing sickle cell disease is not actually being addressed. And while we generally refer to these types of treatments as gene editing, personally, I think they should rather be called gene disrupting, although of course at a very targeted site. And this now finally brings me to the Beam Therapeutics study Beam 102, which is addressing sickle cell disease. And I hope that all my previous explanations and examples given will really enable you to now fully grasp why I believe that Beam have such a unique opportunity and disease treatment modality with their base editing technology. Sickle cell disease is caused by a single point mutation. That means a single base pair in the genetic code is wrong and does not code correctly for the right formation of hemoglobin. And only base editing allows to specifically target and in fact correct that single point mutation. So this means if Beam Therapeutics are successful with the treatment, they are in fact truly addressing and correcting the genetic malformation and therefore truly curing patients with sickle cell disease, rather than going an indirect route through the activation of fetal hemoglobin with all the downsides. As mentioned earlier, prime editing was also invented in Dr. Liu's lab 3 years after the invention of base editing, and this takes us to the year 2019. As I understand it, with prime editing, it is not only possible to change the base pairs C to T and A to G, but in fact it is possible to change all base pairs. In addition, prime editing allows the insertion or the deletion of a group of selected base pairs. And all of this happens without inducing a double-stranded DNA break. And this avoids the complications with the homology-directed repair mechanism, which, as we had discussed earlier, is always prone to errors. And if all of this sounds too complicated, you can think of base editing basically as the eraser. You can delete a single letter and write something new in it, or you can think of prime editing as a search and replace function in your word processor. Both of these technologies allow specifically targeted corrections in the DNA. So, prime editing uses reverse transcriptase in order to facilitate the direct copying of an edited DNA sequence to a specific target site, and this again all without inducing a double-stranded DNA break. From an investment point of view, it is probably worthwhile mentioning that Beam Therapeutics have an exclusive license to the technology that is held by Prime Medicine. So now that we have covered the technological differences between, shall I say, the more traditional gene editing companies versus the newcomers Beam Therapeutics and Prime Medicine, I'd like to just summarize the founding dates and the who is who, the co-founders of the various companies in this overview. So here we have the well-known names Crispr Therapeutics and Editas Medicine, both founded in 2013, Intellia Therapeutics, founded in 2014, and then the newcomers Beam Therapeutics 2017 and Prime Medicine in 2019. Emmanuel Charpentier and Jennifer Doudna were co-founders of Crispr Therapeutics, and Jennifer Doudna was also co-founder of Intellia Therapeutics. The founding of Editas Medicine brought together a lot of the different players in the field, Jennifer Doudna, Feng Zhang, David Liu, as well as Dr. Jiang. And then again, we recognize the familiar names associated with Beam Therapeutics and Prime Medicine. If we take a look where these companies are all located, all of them are located in the Boston area, although technically speaking, Crispr Therapeutics is headquartered in Switzerland, and I presume most likely due to tax reasons. However, all of their research operations, as well as manufacturing and supply for clinical trial purposes, etc., is happening in the greater Boston area. On this map, you see the locations indicated of the companies in the order in which they have listed on the previous slide. And you can see that they're all, geographically speaking, very closely co-located. So to summarize, gene editing is an ever-evolving technology and the companies associated with it. In 2012 or 2013, the CRISPR-Cas system was first described and used to edit cellular DNA ex vivo. And Crispr Therapeutics and Editas Medicine were both founded in the same time frame. As we have clearly seen over this two-part video series, the research into gene editing tools has clearly continued, and prime and base editing have led to the formation of new companies. Clearly, base editing is an evolution of the CRISPR-Cas system and eliminates many of the weaknesses that are associated with the original CRISPR-Cas, which is always inducing a double-stranded DNA break. In order to commercialize this new technology, Beam Therapeutics were founded in 2017. As such, it is probably correct to consider base editing as a CRISPR version 2.0. As always, of course, research does not stop, and the same lab which invented base editing has continued to develop prime editing, and this only within 3 years of the original discovery of base editing. Should we call this perhaps CRISPR 2.1? I'm not sure. I'll let you decide. From an investment point of view, and I have mentioned this earlier already, Beam Therapeutics do have a licensing agreement with Prime Medicine. In my opinion, this is an important aspect when considering potentially to invest in Beam Therapeutics. Regular viewers of my channel know that I focus and pay attention a lot to the clinical pipelines, because as an investor, you really need to know where are the risks, what are the timelines, and the potential markets that can be addressed if successful treatments are brought to market. So, not surprisingly, the older gene editing companies, which were founded in 2013 and 2014, are now well into their phase 1-2 clinical trials. And a newcomer like Beam Therapeutics is naturally expected to be lagging behind. The most advanced programs, Beam 101 and Beam 102, are still near or in the IND-enabling phases, and that means still even before entering into phase 1 clinical trial. Since clinical trials generally are multi-year-long undertakings, it is extremely unlikely and impossible that Beam Therapeutics could potentially catch up in this clinical trial race. So, unless any of the older gene editing companies face any major hurdles and failures in their clinical trials, you can be almost as certain that they would be the first to market with their treatments. We have already seen that there's an overlap in the clinical pipelines between some of the companies, for instance, on their work for sickle cell disease and beta thalassemia. And here I would say it's never that good to be second to market, and I would put really all my money on the Beam 102 study, which, as we have also discussed throughout this particular video, would really constitute the only true treatment of sickle cell disease, rather than going an indirect route of alleviating the disease symptoms through the activation of fetal hemoglobin alone. And if then in the future I was a patient and I had the choice between fetal hemoglobin and if I wanted to exercise, risk being out of breath, etc., I would definitely choose the true treatment of the disease and therefore go with the treatment provided by Beam Therapeutics. And here, if successful, Beam Therapeutics would be the only game in town for a true treatment, and this advantage could likely outweigh being second to the market with a treatment option. Let me know in the comments below what you think. If you believe that you have learned anything valuable in this or my previous video and are able as a result to make better investment decisions, please consider liking and subscribing to my channel. I would also appreciate if you shared this channel with anyone you think who would also be interested in this type of content or investment opportunities. As a thank you, take a look in the description below for how you can access my Gene Editing Pocket Guide entirely for free. Thank you very much.